So we've had light at this point excite this pair of electrons. That is now gone to reduce NADP plus into NADPH. That's sitting there now carrying essentially the chemical energy that was originally electromagnetic energy. Okay? But photosystem 1 is sitting there now missing a pair of electrons, and it's sad, and that sucks. So let's go ahead and make it happy again. Another photon of light is now going to hit photosystem 2. This is happening simultaneously, constantly with this flow of electrons. We're just looking at it kind of one at a time. Okay? And so what's going to happen? Well, photosystem 2 is going to have their pair of electrons get all excited, jump up to a higher energy state, and move over to photosystem 1, because photosystem 1 now is pulling electrons harder since it lost that first pair of electrons that it had. Evolution. There's not necessarily a purpose to it. I mean, the benefit of it that makes something with two photosystems more fit would simply be that now there's more different wavelengths that it can capture. Okay? Yeah. No. So you do not need to know the carriers that are between, but I do. You do not need to know the carriers that are between the two photosystems, but you should know that there are carriers. An important thing about those carriers is that as the electrons are flowing through, what's going to go with them? Hydrogen ions. Hydrogen ions are going to get pumped from the stroma into the thylakoid space. Okay? So those electrons are going to move to a whole bunch of different carriers, and at one point, it's going to attract a proton that then gets pulled to the other side. You don't need to know the exact mechanism. You just need to understand that as electrons are getting shuttled this way, protons are getting shuttled in. Brilliant. So guess what? Photosystem 2 is now sad. Photosystem 1 lost its electrons to NADPH, replenished them from photosystem 2. Photosystem 2 is now sitting there with no electrons. So where is it going to get its electrons? Let's go ahead and take a look back at our picture. What is getting oxidized here? So H2O is what is going to lose its electrons. So there's my water. The hydrogens are just going to stay in the thylakoid space and make that concentration gradient even greater. The electrons are going to replenish those that have been lost. And what is the byproduct of that? Oxygen. Okay. So here we have H2O getting converted into half of an oxygen molecule, because remember, oxygen is stable when it's got two oxygen atoms. We've also got two hydrogens, and then the electrons that eventually go to NADPH. Yeah, Ashley.
Are there only two electrons that are able to be in each photosystem at a time?
There's tons of them, but there are two that are the reactive ones. So, yeah, just think of it as two.
But, like, from water, only those two will go and not all of the electrons from water?
Not at once, but then when another photon of light comes through, the whole thing will happen again. The photosystem 1 loses its electrons, photosystem 2 loses the electrons, and then that other pair of electrons can replenish it. Logan? No. Nope, doesn't matter which one. Yep, it's just constantly moving. Yes. Yeah. So, if we are just tracing one pair of electrons, okay, if we're just tracing one pair of electrons, it's going to start in water, go to photosystem 2, then 1, then NADPH. But the reason I like to do it backwards is so that you can see that this pair of electron is replenished by photosystem 2. So now you understand why there's such a draw from photosystem 2. that it's such a strong electronegativity that it can actually break water apart. Courtney? Yes. Great question. Yeah, the NADPH or NADP plus reductase also uses the hydrogen from out here. So what that means is the stroma is going to lose hydrogens because it's getting attached to the NADPH. The thylakoid space is getting extra hydrogens, both from getting water being split and from getting the hydrogen ions pumped in. So now if we take a look back at our diagram, we've accounted for the water, the oxygen, and the NADPH. The only thing we haven't looked at is ATP, but we're sitting here with all of this potential energy from there being a difference in concentration on the two sides. And so what is that going to cause? Squidward. What is my Squidward over here? That is my ATP synthase. And all of those protons that have now accumulated in the lumen or in the thylakoid space right here, those are going to start flowing out. And as they flow out, what happens to my ATP synthase? Yeah, it literally turns. It's mechanical energy, and as it's spinning, it's going to grab an ADP, it's going to grab a phosphate group, smack them together so hard that they make ATP. My light-dependent reaction is done. Life is good. It's that flow of protons through the middle of it, kind of like a water wheel. So it gets spinning and then can use that mechanical energy. Okay? All right. So I'm going to set up a table quick to compare the electron transport chains in cellular respiration to photosynthesis. I'm going to pause. Okay, so if we're taking a look at the electron source for photosynthesis, where do the electrons come from in photosynthesis? Ultimately, from water. Where do those electrons ultimately end up? Just NADPH. Good. What is the energy source that powers the electron transport chain of photosynthesis? And where does it happen? Raise your hand if you have that and you felt okay about it. Fantastic. Good. Yeah. The NADP plus reductase is, it's kind of like a dock, right? It's kind of like a loading dock. All that's doing is loading the electrons onto the NADPH, but that is like the thing that actually carries them. Okay, switching over to cell respiration. Remember that this is our last step involved in making ATP for cellular respiration. That's our electron transport chain. So what is the electron source of the electron transport chain of cell respiration? It ultimately comes from glucose. That those glucose molecules get broken down. But in the electron transport chain, where are the electrons coming from? If you can't figure out that part out now, come on. Yeah, NADH and? So the electron carriers. The whole point of cell respiration is to break down glucose, fuel these electron carriers so that they can make ATP in the electron transport chain. The final electron acceptor in cell respiration is oxygen, which when it gets the electrons is converted into water. That's literally the exact opposite thing that's happening here. What is the energy source for cell respiration? Is glucose, which then is going to hold that energy in the electron carriers. So that's kind of two answers. And where does electron transport happen for cell respiration? The inner mitochondrial membrane. Okay. When you guys did your video and when you guys did your POGL, there was a lot of reference to all these various sugars that are involved in the Calvin cycle. I'm going to simplify this. What those were really looking for you to understand is that carbon is getting shuffled. But I'm going to go right over here into the stroma. And so I've got this abundance of NADPH and ATP. And I've got a whole bunch of carbon. And really what's happening is CO2 is getting added to another molecule. And then those molecules are getting shuffled around. And then those molecules are getting shuffled around. Okay? Wow, that's super ugly. Doesn't look that bad on my screen. So what is the first phase called when carbon is attached to other things? That is fixation. You do need to know the 3 steps of the Calvin cycle, the 3 general steps. Carbon fixation is first, and there's a molecule that you do need to know that carbon gets attached to. What is that molecule? That is the RUBP. The reason you need to know it is because we have to regenerate it at the end, but we'll get there in a sec. So we've got carbon fixation, which just means carbon is getting added onto this 5-carbon sugar to make a 6-carbon sugar. But you don't even need to know what they look like, nothing. Okay? What's the next step? Well, right now, this 6-carbon sugar is low energy. So it needs to get energy added. What is that called when we're adding electrons and adding energy? Reduction. So the second phase is reduction. Where is that energy coming from? From ATP and NADPH. And that's going to make P-gal. Some of that P-gal is going to be used to make glucose. So P-gal is just a 3-carbon sugar that has energy in it. It's got high energy. So one of those is used to make sugar. 5 of them are used to regenerate RUBP. So you do not need to know every step of the Calvin cycle. You just need to know that carbon is fixed, then reduced, and then RUBP is regenerated. Carbon is fixed, reduced, and RUBP is regenerated. And in each turn, you're making small amounts of P-gal. It actually takes 3 carbon dioxides just to make a P-gal, 6 carbon dioxides just to make 1 glucose. Okay? Actually, that is a lot. I know that.